docking? Like usual, first question, what is docking? Any ideas?
It's like placing another molecule around a target protein.
Yeah. That's pretty much exactly right. So you're placing another molecule around the target, trying to identify the correct conformation that both of them are in, as well as the correct interface that they're actually interacting in. So given two protein subunits, so in this case we're talking about proteins. Of course you can expand this to other molecules, but given two protein subunits, what is the orientation in which they are binding? So we have two subunits. We're just trying to put them together such that it actually makes sense. Just like CASP is a assessment of the community's progress and structure prediction, there's also a assessment for predicted interactions. This is called CAPRI. It's a little less like rigidly structured as CASP. There's not a set schedule for them. CASP is a strict biannual competition. This kind of happens multiple times a year, kind of as we have targets available. This is kind of an illustration of what results were prior to AlphaFold 2. As you can see, for most of the easy targets, which is highlighted by this kind of yellowish highlight, methods were performing all right. We were getting some docs with reasonable quality, so acceptable, medium, or high, and some of the targets even had some nice high quality predictions. However, for most of the hard targets, we were still getting mostly incorrect predictions.
Wait, what's the y-axis here?
Yeah, the y-axis, is .q score, which is really a measure of just docking accuracy. We'll talk about that later, what that actually means. But you can think perfect is a 1 with a perfect aligned interface, all the correct interactions.
So this is protein-protein docking?
Yes. Since AlphaFold 2, as you imagine, we've seen a dramatic increase in the number of successful docks. The easy targets are primarily successful. We have a lot of yellow, red, and green, a lot more green than we did before. We're also seeing a lot of progress in the difficult targets. We're starting to get some acceptable docks, even maybe some mediums, and rarely a high quality dock. So we are seeing a lot of progress in this field as well. One thing I want to highlight is kind of a difference between rigid body docking and flexible docking. So most of these docking methods kind of assume that the proteins themselves are rigid. So they're not going to be changing conformation upon binding or anything like that, which, as we know, may be quite unrealistic. Some proteins are more rigid than others, but in general, this is an unrealistic assumption. However, flexible docking is very difficult because we don't have a lot of data on how these proteins change upon binding. So, for example, we would need two structures, a monomeric structure of the protein as well as the protein in complex with another structure to really have a great understanding of how the conformation changes upon binding. So this is extra data that we need. There's also just a lot of extra degrees of freedom because now instead of just sampling the rigid body transformation between these proteins, which involves a rotation and a translation, we're also sampling all the degrees of freedom of the individual proteins themselves, which just adds a lot of extra complexity. Here's kind of just an illustration of that. So rigid body docking, the protein conformation is exactly the same. They kind of just go together. You're just trying to find how the puzzle pieces fit together. They're not changing shape. However, with flexible binding, flexible docking, the conformation changes primarily usually at the interface, although farther range effects can be seen. So in this case, you can really see this helix, which is more out, kind of bend in. We see these beta sheets kind of open up to kind of give more room for the binding interface. And then there's also this like extra hard problem where we have an intrinsic disordered protein that upon binding forms some sort of structure to form nice interactions around a target protein, this will probably be like the far-reaching like gold standard holy grail of docking. Okay, let's talk about some of the metrics people use for docking. So there's going to be 4 main metrics I talk about here. The first of which is this IRMS, which is just the measure of the interface RMSD. So once we have a proposed docked pose, we look at the interface and compare all the interfacial residues and align that to the true interface, and that's how we calculate the interface RMS, angstroms, and the positive value. Similarly, there's a ligand RMS. So typically when we're doing docking, we refer to the 2 different subunits as a ligand or a receptor. the ligand is typically the smaller of the two proteins, while the receptor is the larger of the two. So the idea is you typically keep the receptor fixed, and then you take the ligand and start throwing it at the target. And then once you have a predicted pose, you just kind of score the RMSD of the ligand itself relative to the target. And yeah, once again, angstroms, positive value. Another interesting metric is FNAT, which is the fraction of native contacts or natural contacts at the interface. So this is a value between 0 and 1 because it's a fraction. And the idea is you really want to have very similar interactions at the interface as the native. So maybe there's some nice salt bridges, maybe there's some nice hydrophobic packing, something like that. So this is just another metric. Early in the CAPRI days, they had like these really kind of ugly, arbitrary like thresholds for each of these to define what was a good quality, what was acceptable, what was a medium quality. This has kind of changed with the development of this .Q score, which is a way of combining all three of these metrics into a same kind of robust single value. And it also agrees quite well with the original classifications of acceptable, medium, and high quality structures that CAPRI originally came up with. So it's just kind of a way of robustly combining the IRMS, the LRMS, and the FNAT into a single scalar value that is a lot easier to compare between structures. There's also like an F1 version of it, which may be a little bit more robust, but yeah, these are the four I wanted to talk about. Any questions?
So, for the fraction of native contact, if the value is 1 for example, would that mean the ligand is covalently bound to the protein because so many contacts?
Not necessarily. So it just means that when we look at the native structure, all of those native contacts, so all of those side chain interactions, all the backbone interactions, are exactly the same in that docked pose. So we have a prediction of what the binding interaction is. We compare that interface to the native interface and see how many contacts are recapitulated.
Basically, all these metrics rely on having a true structure.
Yes.
Docked. Because like if you have PLDDT in structure prediction, you don't really need any reference structure.
Correct. But yeah, we need some like way of measuring how good this prediction is. And yeah, in order to do that, we do need to have the native structure. Although some of the predicted confidences that like AlphaFold is predicting are quite useful for these quality assessment. For example, looking at the LDDT at the interface of a prediction from AlphaFold can really kind of hone in on whether AlphaFold likes that interface, whether it thinks this is a true interface or not, which can be used in like larger scale pipelines to perform maybe more global docking or local docking, etc. Yeah.
Piggybacking off of that, so are there really any predefined or standards when you're performing a blind dock?
Can I defer this to you, Fatima?
Yeah, so what usually happens when you do like make models and stuff is that when you go through the benchmarking process, you try to look at those blind docking predictors like interface PLDT and stuff, correlate it to the known predictors so you get an idea of how well the different metrics work for it. So, and then the other thing that we can do that is kind of always our fail-safe is Rosetta energy, because you really want that stable, like low energy complex. If you have something that's high energy, chances are it's not what you're looking for. So that's always kind of just the fail-safe.
Yeah, and so there's kind of like two sub-problems embedded in the entire docking problem. There's this kind of global search problem of trying to identify the correct epitope on the target protein as well as the ligand protein. So as you can imagine, we can rotate this guy all the way around. So we want to find the actual patch on the surface of the ligand that is interacting. Similarly, we can rotate this guy all the way around and there's a specific patch that's actually doing the interaction. So the idea for the global docking problem is kind of, can you match those two interfaces together? Can you actually find the correct interface? The local docking problem is more of like the actual intricate atomic details around that, so the backbone conformations, et cetera, which is also tricky, but they have different difficulty levels and different things that work better than others. Okay, any final questions? Okay, that's gonna wrap up. Oh, actually, sorry, I keep doing this. There's applications as well. So why do we want to do docking? Well, they can be used to help validate some PPIs. So maybe if you just try to design a binder And if you throw it through the docking method and it recapitulates that binding interaction that you tried to design, that's a really good sign. That's a nice validation that this could work in the lab itself. A lot of these docking methods also include scoring components, so some way to rank poses relative to one another. And this is really useful because it's super easy to generate a bunch of poses, like we can just throw the ligand all around it. But we need some way to differentiate which ones are actually useful, which ones of these are just not useful at all. So a lot of time they have some sort of scoring component, and this can be used to score designs. So once you have something that's already bound or predicted to bind, you can score whether that it thinks that's good or not, but also you can even use it to guide the design process, maybe as an energy or something like that. It's a little bit more tricky to get set up, but it's possible.